Good afternoon, everyone. Welcome to today's webinar, How Oxygen-Intolerant Microbes Navigate an Oxygen-Filled World. I am Tobiloba Oni, a fellow here at Whitehead Institute, where my lab focuses on dissecting the cellular interactions that promote cancer. I am excited to be your host and moderator today. Today's webinar is part of a series. Today's webinar is part of a series of talks that we've organized to share Whitehead community's science and research with the wider community. In a few moments, we'll hear from our presenter, Whitehead Institute Valhalla Fellow Lindsey Backman. First, I would like you to know that this webinar is being recorded for future viewing. Also, we'll be answering questions at the end of today's presentation, but you can submit a question at any time using the Q&A box at the bottom of your screen. With that, I would like to introduce our speaker today, Lindsey Backman. Lindsey began a career at University of Florida, where she earned a Bachelor of Science degree in chemistry. As an undergraduate, she received honors, including the University of Florida Presidential Service and University Scholars Awards. She also participated in HHMI's Exceptional Research Opportunities Program, XROP, and its Capstone Award program, both of which enable students from underrepresented backgrounds to work with a university professor to pursue summer research projects. Through these programs and the MIT Research Program, Lindsey worked for 2 summers as a research assistant in the lab of Catherine Drennan, MIT professor of chemistry and biology and HHMI investigator who subsequently became her PhD advisor. In 2022, Lindsey went on to earn a doctorate in chemistry from MIT before joining Whitehead Institute as a Valhalla Fellow later that year. At Whitehead Institute, Lindsey investigates the structure and biochemistry of proteins in anaerobic bacteria that are abundant in the human microbiome. Specifically, she's exploring the question, how do oxygen-sensitive bacteria within the human microbiome, which are essential to human health, protect the enzyme they rely on for survival? Today, Lindsey will discuss her lab's mission to determine molecular tactics that enable bacteria to cope with varying levels of oxygen and oxidative stress. And she will explain how studying these protective mechanisms could lead to the design of new antibiotics and effective methods of engineering probiotics. Lindsey, welcome and thank you.
Thank you. Let me share my screen. All right. So hi, thank you, Toby, for that introduction. And thank you to all of you for attending and joining us today. My name is Lindsey Backman, and I'm a Valhalla Fellow at the Whitehead Institute. So I'm a relatively new fellow, having had my lab fully open in March earlier this year. I'm really excited to tell you today about my research studying how ancient oxygen-intolerant bacteria, which we also call anaerobes, navigate living in a very oxygen-filled world. So I recently took this photo while visiting the Exploratorium in San Francisco. And this exhibit was called How Oxygen Changed the World. And you can see these 2 beautiful microbial ecosystems side by side. So on the left, we see life before oxygen. We see anaerobes that get energy by metabolizing sulfur-containing molecules and methane. And now on the right, we see life with oxygen. And in this specific culture, we're really seeing cyanobacteria that can actually perform photosynthesis and produce oxygen. And what really fascinates me is that some people might see this exhibit as life in the past, but in reality, the past has really been incorporated into our present. These species still remain in our environments, adapting to a new world with oxygen and so much more. So they can live in extreme environments and what we perceive as normal, even including within ourselves. So first, I'll take you to some of those extreme ones. So bacterial species have adapted all of these unique strategies to thrive under various conditions. So here I'm showing some hot springs. There are thermophilic bacteria and archaea that are able to live extremely hot temperatures, for example, in hot springs. Over here, I'm showing a picture of an oil spill. There are bacteria that are actually able to metabolize oils and pollutants, and they congregate to areas rich in these, to them, nutrients, such as oil spills. So using these bacteria to degrade pollutants have great promise in bioremediation. There are also bacteria in archaea that are able to live in hypersaline lakes, meaning bodies of water that are very high in salt concentration. And they can figure out how to do that in an environment that most species could not live in. And one of my favorite examples I recently learned about is a new species identified, Halomonas titanicae, that was discovered in 2010 from rusticles recovered from the Titanic. So iron oxidizing bacteria by the Titanic actually gain energy to live and grow by transforming iron into essentially rust, iron oxide. Now, the newly discovered bacteria, Halomonas titanicae, are iron-reducing bacteria. They basically eat the rust. So they can reduce iron 3 of rust to soluble iron 2 when oxygen is not available as an electron acceptor. So basically, iron is dissolved. And it's actually predicted that Titanic will be fully disintegrated within the next couple of decades, all because of these bacterial interactions. And I just love this example because it's a really fantastic example of how different bacterial species work in synergy with one another in an interspecies community. They use these unique superpowers to take advantage of nutrients that other species can't take advantage of. And I think that there are so many cases like this in the world, including within us. So all of these, anaerobes that I just told you about and ancient bacteria aren't just around us, they're actually also within us. So through this talk, I'm going to be talking about 2 different types of anaerobes. Obligate anaerobes, or strict anaerobes, can really only grow in oxygen-free environments. Facultative anaerobes, however, can live with or without oxygen. So we don't have to look to these super extreme environments to find bacteria and even archaea. So the most anaerobic environment is actually the gut microbiome. 99% of the gut microbiome are anaerobic bacteria and archaea. But in addition, there are also anaerobes on the oral and genital microbiomes within mucus layers and within plaques. But even, I think, most surprising to me, we have anaerobes in the skin microbiome. For example, C. acnes, which makes up between 80 to 95% of the skin microbiome, is a facultative anaerobe. And as you actually go down into a skin pore, you have a gradient of oxygen. And actually at your follicle, at the subcutaneous follicle, it's actually a relatively anaerobic environment. So again, we really see, we see this gradient of oxygen environments throughout all within us. So one thing I was really surprised to realize when looking at the CDC's list of antibiotic-resistant bacterial threats is that 15 out of 18 of these organisms are either obligate or facultative anaerobes. So both obligate anaerobes like C. diff and facultative anaerobes such as Pseudomonas and Salmonella are pathogens of major interest due to their antibiotic resistance. And some of these bacteria actually switch over to increasingly anaerobic environments as they proliferate and cause infections, as they oftentimes become consumed in mucus layers and form biofilms. So even though these anaerobes are abundant, facultative anaerobes are most commonly studied aerobically outside of animal models. And to me, that means we are missing out on a lot of the chemistry that occurs under anaerobic conditions, and also that in between. So not quite atmospheric levels of oxygen, but also not completely oxygen-free. And so these tactics that anaerobes use to protect themselves amidst low oxygen levels or oxidative stress, I think is a really interesting area to explore for potential therapeutic targets. And I wanted to just bring up kind of a recent news example. You might have heard recently that there have been some spikes of what appear to be mycoplasma infections in both humans and dogs. And as of last week, the World Health Organization officially released a report that they request to have these more thoroughly researched. So first, I'll show the example of dogs. Veterinarians across the country have been noticing what they referred to as a funky pathogen that is causing illnesses in a lot of dogs. And it turns out that researchers discovered it is most likely to be caused by a type of mycoplasma. And they actually think that they said it's similar to a mycoplasma, so it actually might be a new type we haven't seen before. Similarly, you might have heard about these increasing cases of pneumonia in China. And right now, we also do not really understand what is the pathogen causing these, but it's becoming increasingly clear that a lot of these infections are associated with mycoplasma. So what are mycoplasma? So they're referred to as kind of these funky pathogens because they're much different than most bacteria. They actually lack a traditional bacterial cell wall. They're also facultative anaerobes. So what does it mean that they don't have a bacterial cell wall? They actually do not have that traditional cell wall you see. Instead, they just kind of are surrounded by this lipoprotein membrane. There's actually debate for a while of if they could even be considered bacteria, but they are generally understood by the field to be just this very simplified bacterial type. And so because these bacteria, mycoplasma, do not have cell walls like most bacteria do, they actually are naturally resistant to antibiotics that target cell wall synthesis. So between cases like this, where we need to look at antibiotic targets that haven't really been explored before, and when just thinking in general of all the antibiotic-resistant bacteria we have, we really as a field need to start thinking of new pathways and biochemical mechanisms that we can target for therapeutic developments. So how do we even begin to think about a target? So first, I want you to think about how bacteria thrive in a given community. So bacteria employ many strategies to outcompete one another in any given environment. So how do they do this? They can release molecular weapons called toxins. They have the ability, as I described, to survive different harsh environments. They actually can sense and respond to surroundings through a process called quorum sensing, where they can actually release and kind of talk to each other through molecular signals. And lastly, they can consume kind of niche nutrients that other bacteria do not have the ability to profit off of. But today, I'm going to tell you about one type of competitive advantage that my lab is most interested at the moment, the ability to survive harsh environments. And I'm not just interested in any harsh environment. I specifically want to think of exposure to levels of oxygen, varying levels of oxygen, and also high levels of oxidative stress. And so we've talked about how these microbes really rely on oxygen-sensitive pathways to live. Their basic metabolism relies on some of these enzymes that can't even be exposed to oxygen momentarily. So now I want to add in an additional caveat. When you think of an infection or cancer, so in the case of the gut, perhaps colon cancer, or you think of inflammatory bowel disease or Crohn's disease, all of these disease states trigger inflammation. And inflammation leads to a host response that is associated with high levels of oxidative stress. Now, these microbes are so sensitive that they're vulnerable just to molecular oxygen, much less even more highly oxidizing agents that are formed, such as different reactive oxygen species. So I want to know, how do these bacteria actually protect these pathways when exposed to oxidative stress and disease states? So overall, that is the Backman Lab's goal right now, to determine the chemical tactics that allow these anaerobes in the human microbiome to cope with varying levels of oxygen and also high levels of oxidative stress in disease states. I just want to give a shout out to this illustration by Caitlin Rush that I just love, that she designed for a talk recently. So to probe these questions, I first want to introduce you to some equipment that I'm really excited about. So here I'm showing you an anaerobic chamber that we have set up. So this chamber, while within this glove bag, it is completely under a nitrogen and hydrogen environment. So it actually has a palladium catalyst within it that removes any trace oxygen. If oxygen gets in, it reacts with the hydrogen with that catalyst. And so to regenerate our catalyst, we just take that catalyst out and essentially evaporate off the water. We also really are, we designed this anaerobic chamber to be specifically for bacterial culture. So we have an incubator in it that can range up to 65 degrees Celsius for our culturing experiments. We have a dehumidifying system that's really important for bacterial culture, hydrogen sulfide removal column, which is going to allow us to really study a range of bacteria, since so many anaerobes actually produce hydrogen sulfide. And what I'm actually most excited about is this subchamber that actually enables infusion of controlled amounts of oxygen to small scale bacterial cultures. And we are about to get a 24-well probe that actually allows us to measure the oxygen level and pH in every well. So we can really do controlled experiments studying that in between I talked about. So somewhere in between purely anaerobic and atmospheric levels of oxygen. So what are we going to do with this subchamber? So what we would like to do first is start basically screening for pathways that offer organisms with protection against oxygen. So to do that, we're going to be using different screens using both a CRISPR interference or CRISPR-I approach that will be working with Heather Keyes in the functional genomics platform, and also some RNA-seq approaches. And another thing that's really cool about this system is that it can also be used to co-culture host cells with host cells, meaning like mammalian cells, with bacterial cells. And this is more so future directions, but I'm excited about the possibility that we have with this instrumentation to actually start probing host microbial interactions. So right now I want to focus on one possible strategy for protecting oxygen-sensitive enzymes against oxidative stress by actually physically shielding these pathways. So that pathway is basically, strategy is the use of bacterial microcompartments. So these are really giant, essentially bacterial organelles that physically encapsulate oxygen-sensitive pathways and might actually act as a shield to protect those enzymes. So I want to take us back to kind of when you first maybe learned about prokaryotes and eukaryotes. So traditionally, in a biology textbook, they present prokaryotic, eukaryotic cells in this manner. And honestly, I'm guilty of this myself, as I just showed you that very simple picture of mycoplasma. But traditionally, how we see prokaryotes, is that they're very small and simple. So they don't have all these fancy organelles. They're basically just have, this clump of that we call nucleoid, a bunch of DNA smushed together, some ribosomes sprinkled around, some plasmas. They have a cell wall most of the time. But for the most part, they're pretty simple. There's a lot of beauty in that simplicity, but I think we might have simplified it a little bit too much. On the other hand, eukaryotes are big and complex. They have the organelles. They compartmentalize their different machineries. And so if there's one thing I want you to take home with you today regarding what we're learning about bacteria is that they're more complex than we thought. So we're realizing that there are more and more examples of organelles in bacteria. So when I say organelle, I mean basically a way of compartmentalizing machinery, cellular machinery together. And we're finding examples of both protein-bound and lipid-bound organelles in bacteria. And this is actually kind of a paradigm shift in bacteriology field. And it's rapidly evolving. We're discovering more and more new ones. And I'm very excited to start getting into this field. So if you have heard of a bacterial microcompartment, the one that is most well studied are called carboxysomes. So carboxysomes have been studied for decades. And basically, these are involved with carbon fixation in photosynthetic bacteria. So they are associated with the enzymes rubisco and carbonic anhydrase. Here you see these really beautiful purified BMCs. BMCs can actually vary from between 50 even up to 900. 50 to 900 nanometers, which is really gigantic in when you think on a microscopic scale. And it was always proposed that carboxysomes might allow for the substrate CO2 to reach a critical concentration for catalysis and reduce the entry of oxygen. And recently, the carboxysome field has done some very beautiful structural work using cryogenic electron tomography, where you can actually see what's going on within these bacterial microcompartments. So here I'm showing basically a slice of a cell, and what we can see within it, each of these kind of hexagonal units is a slice of the carboxysome. And you can see these enzymes actually forming these fibrils. And they also found that carbonic anhydrase, the other enzyme, comes and directly interacts with these long rubiscal fibrils. So you're seeing these really beautiful large-scale macromolecular structures. And I'm excited to do those kinds of studies with the bacterial microcompartments that we are interested in. So even though carboxysomes are the BMCs that are most well characterized, they're actually not the most abundant BMCs, at least based on bioinformatics studies. So based on bioinformatics studies, the enzymes most commonly associated with BMCs are B12-dependent enzymes, so vitamin B12-dependent enzymes, and what we call glyceradical enzymes. Glyceradical enzymes are so oxygen sensitive that if they are exposed to oxygen, the protein physically breaks into two pieces and cannot work anymore. So these by bioinformatics are actually the most common BMCs. So how do these work? They basically take in a substrate and they perform all of the a whole metabolic pathway within the BMC, and then they release specific products. So you can imagine that having all of your oxygen sensitive enzymes really stuck and shielded by this protein layer could potentially restrict the amount of oxygen and also different forms of reactive oxygen species from entering into this microcompartment. But there's still so many open questions in this field regarding the basic biology. First of all, it's really been proposed that BMCs protect against oxygen, just because they're generally associated with oxygen-sensitive proteins. But nobody has studied that. And so do they actually offer oxygen protection? That is an experiment that we need to do, and our lab is really equipped to do those experiments. Secondly, these encapsulated enzymes have proposed and characterized auto-encapsulation domains. So what does that mean? So when you're thinking of a protein, they actually have an extra part of the protein that somehow allows this protein to get inside this organelle or BMC. But we don't know how that process works, although I would think that we have a little bit of a clue that we're exploring. So right now, the BMC we are looking at are, I'm going to refer to them as the UT BMCs. That stands for ethanolamine utilizing BMCs, and they require vitamin B12. So these BMCs take in the nutrient ethanolamine that is super abundant in the diet and also in gut epithelial cells. And so what they can do is they take in ethanolamine, they undergo many chemical reactions, including some oxygen-sensitive proteins, and then they release products out of the BMC that are involved with just basic metabolism. So the current aims we've been going after so far are just optimizing how we form and purify the EUT BMCs in E. coli for our biochemical and structural experiments. And we're also really focused on structurally understanding how these enzymes get into the BMCs with these proposed auto-encapsulation domains. So how do we actually isolate a BMC? What we do is we basically take bacteria that we culture on these plates and we inoculate them overnight in just a traditional LB media. In microbiology, that's the typical bacterial media that is used. Then we need to transfer that to a new type of media to actually induce the formation of BMCs. And this media includes the substrate, ethanolamine, and vitamin B12. And so I often get asked the question, how did we go so long not seeing these bacterial microcompartments, these giant organelles, if they are so abundant and large? And I think that this step right here might be the reason why. In microbiology, at least with studying bacteria in culture, we, as a field, have generally used some kind of standard approaches. So in order to form the BMCs, if we just put them in LB media, they don't actually grow BMCs. You have to give them a high amount of that substrate ethanolamine and B12. Now, ethanolamine within a host, such as ourselves, is super abundant. And so sometimes what we observe in just an LB culture isn't actually close enough to the conditions that lead to the phenomenon that actually occur within us in all different kinds of environments. So I think that's something just to keep in mind in microbiology. So our initial hurdle was that some of these E. coli lab strains really did not grow under these BMC forming conditions. So Nina Greeley and Dawson, so Nina was an MSRP student in the lab over the summer, really figured out, they screened a bunch of different E. coli strains that encoded only this EUT-BMC, and they actually identified 14 E. coli strains that grow under these conditions. And now in MIT Europe, Aidan Bousquet is taking on this project with me for really optimizing our growth and purification of these BMCs. So how you actually then isolate them is through centrifugation. These are so big that you can actually pellet down, lyse the cells, pellet down kind of all those membranes, take the soluble fraction, and then go to centrifuge at a high speed, and your BMCs will actually pellet out. And so here I'm just showing that in our initial experiments, we do get we observe proteins at the some of the band lengths that you would expect for BMCs, but this is still definitely a work in progress. So what are some future experiments we want to do with this? So we want to start performing these BMC growths under different levels of oxygen. There are some protein factors that are thought to only be associated anaerobically versus aerobically. They also really vary in size and abundance. So I'm excited to start exploring those differences. We want to do some electron microscopy studies to actually structurally characterize BMCs, similar to those really beautiful structural studies that you saw with carboxysomes. We want to identify the different protein factors that are involved with BMC formation, degradation, and just stability through some CRISPR interference screens and also proteomics experiments. And then lastly, we eventually want to do some genetic knockout experiments to basically see, if you have a bacteria that with this metabolic pathway and you have one that has these shells, that physical shield, and some that don't, do the ones with the shield, when exposed to oxygen, actually have a competitive advantage? So for next, I want to kind of take you towards how we're thinking about how these enzymes actually get in the BMCs themselves. So I'm going to show you a little bit of structural biology. So this is kind of my bread and butter. So crystal structures have been solved for these ethanolamine ammonium lyses. So basically the enzymes associated with the UBMCs. But these are not actually enzymes that are in organisms that have that bacterial shell. So I've done some predictive analysis using AlphaFold to look at this additional domain on the N-terminus. And this additional N-terminal tail is thought to be involved with auto-encapsulation. So somehow having these little extra pieces at the tail of the protein allows them to actually get into this BMC. But we don't really know how yet. So the first thing we were wanted to do was we wanted to structurally characterize this auto-encapsulation domain, as nobody has done that so far. So over the summer, Nina and now Aiden will be overexpressing and purifying the subunits UB and UC, so enzymes that are involved that do actually have that auto-encapsulation domain. And so far, we have some crystal hits. So a very common structural technique we use is called X-ray crystallography. You basically figure out how to give your protein the magic solution that will allow them to form a protein crystal. We can shoot X-rays at those crystals, and based on how they diffract, we can then back solve what that structure of the proteins had to have looked like to get that pattern. So we can solve an experimental protein structure like I showed you before. These are kind of fun. They're pink because B12 is pink. If you've taken a B12 supplement, you probably know that. But this is really exciting because this means we're on the way to solving structures of enzymes that have these domains that allow them to be encapsulated into a BMC. I'm also really interested in what does that N-terminal domain work with? And so based on some hypotheses in carboxysomes, I found a similar protein with the EUT pathway that basically, I think, acts as a tether. So US is one of the shell proteins. It's you can think of them as one of those little pieces on this giant icosahedral unit. But then it has two really floppy ends. And it's been thought with carboxysomes that these floppy ends basically tether the N-terminal auto-encapsulation domains, and then allow, kind of brings them into the BMC. So again, I used basically a prediction model with AlphaFold multimer of how UB, C, and US might interact. And we do see some interactions potentially between that kind of flexible loop and a UB. This is not, I'm not super convinced by this, but it is, I think, a starting place. And I now want to follow up with some cryo-EM structural studies to actually try to get an idea of how US might recruit other enzymatic factors into our bacterial microcompartments. So all of this work was done in E. coli, but eventually I want to expand into other organisms. And I think the first one I want to look at is Salmonella enterica, specifically the ones associated with typhoid. So why Salmonella typhimurium? So this is a pathogen that is found in the gut, so an extremely anaerobic environment, but it's known to be particularly resistant to large bursts of oxidative stress. So it can use propanediol and ethanolamine as their sole carbon sources, so they're kind of sole nutrients. And they actually also encode bacterial microcompartments for both of these metabolic pathways. And there have been some studies in mice that actually show that these BMC operons are upregulated upon infection in mice, and that addition of those substrates, propanediol or ethanolamine, results in substantial growth advantage and virulence. And so I think that this is a kind of a fun example to look after. I don't know if fun is the right word, but intriguing because this unique super ability to be able to resist oxidative bursts and oxidative stress could give us some, keys into how bacteria do this. And, in the meantime, could really give us some potential new antibiotic targets for this antibiotic resistant bacterium. Lastly, a question we're really interested in are, do these BMCs actually even give a competitive advantage? So the way we're going to do this is we're going to take a genetics approach. So we're going to take strains of E. coli and Salmonella, basically some that contain those BMC shell proteins and some that don't, because actually a lot of strains do not have the shell proteins, they just have the metabolic pathway. And so we're then going to culture these under various controlled oxygen levels and basically measure their relative abundance by flow cytometry. And so what we're looking for is, when you're exposed to oxygen and you have this protective shell, does it actually allow these bacteria to grow more robustly under oxygen than these that do not have that added protection? And then I'm just going to end to give a little bit more of an overview of other types of projects our lab is going to be going towards. So BMCs might also play roles in regions other than the gut microbiome. So this could include the skin microbiome. So C. acnes is a facultative anaerobe that most people makes up 90 to 95% of the human skin microbiome. Different strains actually have really varying oxygen tolerances. So C. acnes is known for its ability to metabolize propanediol. Actually, its first original name was named specifically after that ability to degrade, to metabolize propanediol. And that pathway is actually associated with a bacterial microcompartment. And so I'd really like to explore the role that this BMC has in C. acnes and whether it confers any kind of oxidative protection, and also screen for other protection pathways within C. acnes. So overall, what can we gain from this? So by understanding these new oxygen protection mechanisms in our bacteria, perhaps these BMCs or perhaps other mechanisms that we discover through these different types of screening approaches we will be taking, we might be able to design new drug targets towards these new pathways, especially since I think targeting specifically anaerobic pathways is not something that has been thoroughly explored yet. Second of all, for whatever reason, a lot of pathogens tend to be a little bit more resistant and have more strategies than commensal or those good bacteria have within us. And in order for a new bacteria to be introduced and to actually stick in a microbiome, so with a probiotic, they need some type of competitive advantage. And there's a lot of research that shows that. So if we can determine what some of these competitive advantages are, we could maybe engineer new probiotics that could provide that competitive advantage. And lastly, I brought up at the beginning how there's a lot of promise for using oxygen-sensitive enzymes, for example, their abilities to degrade pollutants and oils for bioremediation and industrial uses. But one of the biggest issues is that they're usually pretty oxygen resistant. So figuring out how we can make these bacteria less oxygen resistant could potentially make it more viable that we could actually use them in in environmental ways. So overall, our lab really just aims to kind of gain some insight into how these various oxidative states, whether it's different exposures to oxygen or actual oxidative stress, impact anaerobic microbial physiology, and how that in turn impacts us within both our study and diseased states. So with that, I just would like to thank our lab. Over here, you can see we made some microbe shoes and microbe bags. It was a very fun lab activity. I'd like to thank our whole lab, Professor Deb Hung over at Broad MGH, who I learned some bacterial genetics with in the first 6 months as a fellow, some collaborators, Tammy Lieberman and Mike Laub, Heather Keys from the Functional Genomics Core, mentors, Professor Ruth Lehman, Sebastian Laredo, and Jonathan Weisman, cores, and funding. And with that, I am happy to take any questions.
Fantastic. That was very great. So we have several questions here, but also if you have questions, please put it in the Q&A box. So the first question is from, it's actually not clear who actually sent the question, but the question is, would it be informative to do an experiment in which you express BMCs in anaerobic bacteria without BMCs to see if this gives them an advantage in high oxygen conditions?
Yeah, so I think this person is kind of proposing the opposite of that last experiment I said, of where we basically take bacteria that do have those BMCs and then knock it out. And I think that is a great idea, that you could also do the opposite of knocking it in. So these metabolic pathways are are conserved across many species. Some have BMCs, some don't. And one thing I didn't say is actually BMCs are even more prominent in pathogens than they are in commensal bacteria. And so you do wonder that, yeah, if you took, a commensal strain and engineered those shell proteins in the same operon so that they're regulated together, could you actually, form these BMCs and then do that exact type of competition study to figure out if there's competitive advantage? I think that's a really interesting study. And I will say that there has been success of people engineering these BMCs, not just, actually really interestingly, so the engineering field has actually advanced even faster than us understanding the basic biology of bacterial microcompartments. So there have been studies of actually engineering BMCs, so these shell proteins, around enzymes not even associated with BMCs. So iron-iron hydrogenases, nickel-iron hydrogenases. So just to see if you could form these on any other metabolic pathway just by having that mysterious auto-encapsulation domain. And it works. You actually can encapsulate them. And so I think that's a great question that could get right at that question of, is it actually giving it advantage? And specifically, is it giving it advantage because of that oxygen exposure?
Wonderful. Another question is, does adaptation to low oxygen increase health or lifespan in any species?
Yeah. Okay. So to answer this one, okay. So when we think about lifespan in bacteria, it's a very different, I would say a different term or way of thinking, because bacteria are single cells and they're constantly dividing. And so when we think of them in the microbiome, you're not really thinking the lifespan of one single microbe. You're thinking of a microbial community, and basically having enough of that community develop a niche, a specific part of the microbiome, and have enough of a competitive advantage to stick around. And once a bacterial species has done that, they're usually pretty good at sticking around unless there is some kind of disturbance. So, okay, now I have to get back to the question. How do oxygen-intolerant microbes tolerate and increase health? So I wouldn't say that it necessarily increases their lifespan. I would say, just in some ways, it increases their health of bacteria by being more robust. They have more options of environments they can live under. And so I think in that way, it does give them increased health or an increased advantage, because they're able to adapt and tune their metabolism to live, in states where they are exposed to oxygen, and then they aren't, and then they are exposed to oxidative stress, and then they aren't. So it makes them more robust, I would say.
Is there evidence of BMCs in obligate anaerobes such as Bacteroidetes?
So as far as I know, these are largely associated with facultative anaerobes. So I have not, as of now, I have not in bioinformatics seen strict anaerobes that have these BMCs, but it doesn't necessarily mean that they don't exist. I will say that it's just really interesting, like, when I applied for the fellow's position and was really thinking about these BMCs, there were like 7 main BMCs that were out there. The last I looked, like a month ago, there are now, I think, 18 different types of BMCs that exist. We're constantly learning of more of these types of BMCs associated with pathways, different shell proteins and shell patterns that all tend to make this icosahedral unit with different patterns, which is cool. But yeah, I'm not going to say they don't exist, but they have not been discovered yet.
Yeah, so another question is, oxygen and ROS are small. Does structural modeling support the hypothesis that BMCs would exclude this species?
It is a great question. And that is why we need experimental evidence so badly. You're right. And honestly, even thinking back on the carboxysome field, thinking about how CO2 might be able to get into a shell, but O2 can't. is a little, if that's the case, that's amazing. And so it would really rely on, I think, probably the electrostatics of these pores. So you basically have these shells that really fit together, and then you have selected pores. And so the pores vary based on shape and electrostatics to allow whatever substrate you need. But yeah, I also don't know if maybe it does let in oxygen, but you just have less exposure than if it were just free and in the open. And so there could be a competitive advantage without being so sharply restricted. And so, yeah, that is a fantastic question. And I think, so I was at the ASBMB, so the American Society for Biochem and Molecular Bio, and they actually had a whole session on BMCs for the first time, which I was really excited about. They called it bacterial organelles. I was like, yes, bacterial organelles. And I mentioned that we were studying this, and they were like, thank goodness, because we have all these people who are engineering BMCs for oxygen-sensitive enzymes. But nobody's actually studied the protection it adds if it does. What we know is that we've only discovered BMCs that protect oxygen vulnerable enzymatic pathways.
So another question quite interesting is, are eukaryotic B12 utilizing pathways cytosolic or mitochondrial? Or they're also known to phase separate. So again, are eukaryotic B12 utilizing pathways cytosolic or mitochondrial, also known to phase separate?
So eukaryotic ethanolamine. Wait, sorry, do you mind?
Yeah, do they phase separate? Phase separation?
So the phase separation question is definitely something I want to look into. I would be really surprised if they don't phase separate to some degree. It's really funny when I was first learned about how in carboxysomes they have these kind of shell proteins that have these flexible tails that congregate things together. They called that phase separation. And I was like, I don't know if that's phase separation, but it would really make sense that these do need to phase separate in such a way that that whole metabolic pathway can be encapsulated. I think I'm a little confused with as far as the eukaryotic ones go, just because those metabolic pathways are very different. They are cytosolic metabolic pathways, but they are not associated with BMCs. And I'm honestly not sure. If those how many enzymes are in that pathway and if they actually do phase separate. But it's a much different type of chemistry. The types of chemistries that are being performed in bacteria, eukaryotes don't have those enzymes. So
yeah, there's one that I find actually quite interesting because it applies also in the case of cancer. It says, how do invasive anaerobic bacteria survive in the host cell? Do they localize in specific compartments or alter the cellular oxygen environment?
So most of the bacteria that I'm talking about are not intracellular pathogens. And so at least, the ones that we are studying currently, like, E. coli, Salmonella, C. diff, all these ones in the guts, they don't actually invade the host cell. So they are kind of, when you think about it in the human gut microbiome, you basically have a lining that has to be really, you have to have a really good lining, because basically you have this lining that then protects from, so an epithelial layer that protects from your bloodstream. And that really needs to be preserved. It has lots of layers of, collagen, epithelial cells, etc. But on the other side is your bloodstream. So that is when, if you have really severe bacterial diseases that are not actually cured, oftentimes death is by sepsis, because it eventually gets into the bloodstream. And so as far as compartmentalizing in the cell, I'll be honest, there are definitely bacterial pathogens that are intracellular. I don't really know where they compartmentalize, and actually if they have these BMCs. there are a lot of great people on campus, like Professor Becky Lamison, who could probably answer those questions better than I can. But I do think overall, the redox environment can really change based on the general site. So for example, if you have a C. diff infection, it's going to be those epithelial cells become highly inflammatory, and actually the bacteria start degrading the collagen and all these protective layers to kind of invade, and that's how you get disease progression. So yeah, usually when I'm thinking of bacteria, I'm thinking of them interfacing between two cells, not so much one being encapsulated in the other.
And actually, speaking of encapsulation, I mean, since mitochondria is essentially a derivative of bacteria, there's a question here that says, do mitochondria make use of BMCs to compartmentalize reactive metabolic intermediates?
Wait, can you repeat that one more time?
Do mitochondria make use of BMCs to compartmentalize reactive metabolic intermediates? Do they have it?
I mean, it's a great question. If you think of in just biology, of what we know of in biology right now, the closest physical structure we really see to a bacterial microcompartment are viral capsids. They're these really beautiful icosahedral units, and I didn't really get into it too much, but every different type of BMC is composed of between 3 to, I think, up to 9 shell components that are basically form this beautiful mosaic that repeats to form this icosahedral unit. I don't know of cases in eukaryotic cells that have these types of structural units. That being said, I see a lot of parallels between phase separation and BMCs. That this is another way of how you can get all, these complex long metabolic species all clustered together. And so why they didn't evolve into eukaryotes, if they didn't, they might exist and we just, have not found them. But it's a great question. I don't know.
There's another one here that asks whether the environment of facultative anaerobes affect BMC. For example, anaerobes that infect the host versus free-floating aqueous anaerobes.
Sorry, what was that? Free-floating what? Anaerobes?
Aqueous anaerobes. Like essentially, if you have an anaerobe, invading or infecting some host, or if they're just free-floating.
Okay, yeah. So I do think being associated with the host definitely would likely lead to these BMCs being formed. Because the way you have to think about it, I mean, if you notice some of those pictures, like you look at the cell and so the cell you're thinking about 6 microns at max, like the ones that we're studying are like 1 to 6 microns. A BMC that can get up to they're generally 100 nanometers, but those are large proportions of the cell. And I brought up mycoplasma at the beginning. They do have encode for BMCs. Those are even smaller. Mycoplasma are the tiniest bacteria that we know of. And so there's a large expense that you have to think about of making this organelle that is taking up such a huge portion of your cell. And so I don't think they would grow unless they have enough of the food, essentially, these substrates in what we call cofactors like B12 to induce formation. And so that is still really an unexplored area, which is why I want to do these different conditions of oxygen, pH, things like that, because when I say that they range in these ways, it's like one person does a set of experiments and their BMCs tend to be about this size. And then, you do it another one and they're a different size. And so what are those variables that actually impact how many of these make, and the size of them is really interesting, especially because when you think of size, it's literally a repeating mosaic unit. And so it's not like something that can just expand and stretch. You need more repeating units for it to grow. So it actually has to be quite controlled to end up with an icosahedral unit. And so it's a really fascinating field. But I would say that unless they are free floating in an environment that has those specific nutrients, they likely would not form them because it's such a high energy burden. But in the case of the human gut, and especially for a pathogen, you're looking for some kind of competitive advantage. And especially if you're an infection or, bacteria that are in an inflammatory environment, you might do anything you can do to get that leg up. So I mentioned that it might protect against oxygen. Another proposed benefit is that they actually encapsulate toxic intermediates within the shell and only release the product. So that could be another advantage, that your cells are actually more viable and therefore, reproducing and cultivating this like healthy community over other bacteria around. So
I see. And I'll ask the last question, which is more general. I would like to know, Lindsey, are there ways in which we can actually leverage these BMCs for some, biotechnological function or use? Are you thinking about that? And what are you thinking about in terms of that?
Yeah. So as far as leveraging this, aside from like thinking of ways, if we do find a competitive advantage, I think disturbing the shell would be a kind of a natural direction, using therapeutics to disturb the shell. I'm really interested in probiotics. I think that if you could take, I'll give I have 3 minutes left. So one of my favorite papers of all time is from Justin Sonnenberg's lab. They basically took commensal bacteria in mice, and they tried to basically give it to the mouses. It's a typical probiotic. Goes straight through the mouse's system, doesn't stick. Then they engineered it to be able to metabolize a unique nutrient found in red algae. They gave that bacterial strain, goes through, doesn't stick. They then gave that bacteria to the mouse with red algae, and it stuck in the microbiome. And then they also gave it that strain and the metabolite, that specific metabolite that it can use in its metabolism, and it stuck. So literally just having one competitive advantage gave it enough of a niche to actually engraft in the human microbiome. And so I think that is actually a pretty remarkable idea for being able to advance probiotics, is thinking of what are those one advantages that we can make stick. So if we can give, somebody suggested, engineer BMCs around a shell of something that is known to really be associated with health, that might be a probiotic that could stick. Furthermore, what if you could isolate BMCs that have some of these oxygen sensitive proteins that can degrade oils and pollutants? You actually engineer those into bacteria, and then if you have an oil spill, actually introduce bacterial cultures into them. And so because those enzymes are actually, they're oxygen sensitive but not associated with the BMC. And so I think those types of engineering experiments could really be leveraged to not only address, antibiotic therapeutics, but also think about engineering effective probiotics. Since right now, like, for example, C. diff in the worst of infections, the best treatment we have are fecal transplants, which is essentially just a probiotic that works. So if we could develop fancier probiotics that really work in these diseased cases to reestablish a healthy microbial environment, I think that would be amazing. And also, like I said, the capacity to leverage these against environmental issues, I would be really excited about. So.
Thank you so much. That concludes today's presentation. Thank you all for joining us today. And special thanks to Lindsey for sharing your work with us. Take care.